Hello everyone. So in this video we are going to talk about induction, which is a simple process in developmental biology. So let's start with its introduction and the definition of induction. So induction. Signal induction refers to a cascade of signaling events during which a cell or tissue signals to another cell or tissue to influence its developmental fate. Now for example, if there is a group of cells which produces some chemical factors or growth factors or differentiating factors that goes to its another group of cells or tissues and produces some changes in its or influences its developmental fate. So if these cells produces some growth factors or differentiating factors that bring some changes in another group of cells or tissues, so that process is known as induction, right? So there are two components of inductive interaction. Two components: first is inducer, that brings the change, and responder, which responds with changes. Responder are those cells which changes. So that's about inducer and responder and induction. So now to understand this concept a bit more, we have to know about the development of frog embryogeny, right? So for example, if this is a frog egg, this is its animal pole, this is vegetal hemisphere, animal hemisphere, and here the sperm comes and fertilizes the egg, right? So this black point is the point of sperm nucleus entry. So just 180 degree to the point of sperm nucleus entry, a gray crescent, a gray area or gray crescent is formed. This is a very important part of the egg for the future development of the frog. This gray crescent, you know how. And the first cleavage happens like this from the mid of that gray crescent. So if this is a frog embryo, this is a gray crescent, and the first cleavage occurs something like this. So we would know what this gray crescent, why this gray crescent is so important. Gray crescent, let's see. So to tell us what is induction, Rund and Spemann did an experiment to emphasize the vital role of gray crescent in amphibian development. So you can see if this is a frog egg and this little area is gray crescent. So what happens? First cleavage occurs so that the gray crescent is divided into two equal parts. If such a cleavage occurs by dividing two equal parts of gray crescent, so two normal frog embryos are formed. But if the gray crescent is only found on one side of the cleavage and the other side has no gray crescent, then what would happen? The part with no gray crescent would not be able to form the dorsal side of the embryo. So only a belly piece of undifferentiated cells will be formed, while the part with the gray crescent will form a normal functioning tadpole, right? So if the egg is surgically divided into two halves, right? So egg is divided into two halves, each having one of the gray crescent, one half of the gray crescent. Each half of the egg will develop into an entire embryo if cultured in isolation. So what they are doing is, if this is a egg, a fertilized egg with gray crescent, so they surgically divide the embryo into two parts, right? And if every part have equal amount of gray crescent on either side, then two normal tadpoles are formed, right? So this was the experiment of Rund and Spemann. But if the same operation is performed on an egg in which one half gets the entire gray crescent while the other blastomer lack the gray crescent material, and the two blastomers, when cultured in isolation, the blastomer which is developed devoid of gray crescent will develop a simple sac of ectoderm containing endoderm, while the blastoma having the gray crescent will form an entire embryo. So what is saying that if you do this, that if you surgically divide the embryo, one part have the gray crescent, this one has gray crescent, this one have no gray crescent. So the piece with no gray crescent will not be able to form a normal embryo or a normal tadpole. It will form a belly piece with only endoderm tissues, with no tadpolic characters, while the piece with the gray crescent will form a normal tadpole and a normal development. So this was the experiment of Spearman and Rund. So why this happens? The vital dye method and other marking devices such as the carbon particle technique and radioactive labeling method have been applied to ascertain the fate of gray crescent during development. So what we did to find out why this gray crescent is so important for the development of an embryo, what they did, they labeled radioactively the gray crescent area, and they saw what the gray crescent forms, right, in an embryo, what the gray crescent forms. So these studies revealed that the gray crescent material of the egg gives rise to dorsal lip of the blastopore. So gray crescent forms the dorsal lip of the embryo. Now we will see why this dorsal lip is so important for the development of embryo, but for now all the important thing is that gray crescent forms the dorsal lip of the embryo. So the dorsal lip of blastopore and its significance. Let's see. In normal amphibian development, the dorsal ectoderm just above the dorsal lip of the blastopore becomes the neural structure. So it's saying that if this is the dorsal crescent or gray crescent, what you call, right? This is the dorsal, the gray crescent area. So the part just above the dorsal lip, if this is the dorsal blastopore lip after invagination, so just the part above the dorsal lip for the neural plate, this red area forms a neural plate, right? The dorsal lip region is called chorda mesoderm because it develops normally into mesoderm and notochord, while this blue area, which is the dorsal blastopore lip, it forms the notochord. So this happens by the induction. Induction is what? Induction is the process by which a group of cell or tissues induces some changes into another group of cells or tissues, right? So here this dorsal lip region induces changes in its above tissues to form a neural plate, right? So this dorsal lip region is an inducer which induces to form the neural plate of the embryo, right? So let's see how they proved it. So what they did, this Spearman and his student Mangold, by using a transplantation technique, have shown that the dorsal lip of the blastopore, when implanted to the ectoderm area of the same or another embryo at the stage of development, the dorsal lip is able to initiate the formation of secondary embryonic axis. So how they prove that it is the dorsal lip that forms the head region or the neural region or the primary axis of the embryo. How they did this? What they did was they took a glass scalpel and cut out the dorsal blastopore lip from one embryo. They cut it out and they transplanted that dorsal blastopore lip into another embryo that already has a dorsal blastopore lip on this side, and they took it out and transplanted another dorsal blastopore lip on another side of the embryo. So what happened with two blastopore lips on that embryo? Two primary neural plate appeared. That is, two heads on the same embryo occurred. So something like this was formed, induced double body, right? So it was then hence proved that what the dorsal lip does, it is an inducer, dorsal lip an inducer of forming the neural plate or the primary organizer axis, right? So dorsal lip is an inducer What it does, these dorsal lip cells induces to the formation of the neural plate. So what happens? Let's review this experiment once again of Spearman Mangold organizer. So what they did was that they cut out the dorsal plastopore lip from one embryo, and they gave it to a donor, to receiver embryo, another embryo. So this receiver now has two blastopore lips, right? Two blastopore lips, and when that happens, we know that blastopore lip induces the formation of neural plate. Neural plate. So the dorsal blastopore lip induces the formation of neural plate. So when an gastrula have two blastopore lip, it induces the formation of two neural plates. So two neural plates are formed, right? So two heads of the um, same tadpole occurs. So this is a strange kind of experiment, but it worked to demonstrate that the blastopore lip is an inducer. Let's see this. So Hans Peyman discovered organizer. So what was the experiment? Transplantation of different parts of the developing embryo donor into another embryo host. So result, if the dorsal lip is transplanted, a second embryonic axis or head is formed. Right. So this was the experiment. Now embryonic induction is a process whereby a cell group causes another group of cells to differentiate to a particular developmental fate, right? That's what we saw it earlier. That's what we saw in this experiment, that the dorsal blastopore lip induces other cells to form neural plate. That's what it is. Embryonic induction is a process where a cell group causes another group of cells to differentiate to a particular developmental fate. Such a phenomena is the recurring process in embryonic development.